We live in an era where DNA provides the definitive answers for forensic analysis, paternity testing, and genetic screening. It is the gold standard for proving biological identity. Because these applications are so common, it is easy to forget that less than a century ago, the most sophisticated scientific labs on Earth were still operating in total ignorance. They had no idea that DNA was the carrier of our hereditary information. Biologists understood the effects of inheritance. They could see how specific traits were passed through generations of plants and animals. But the physical molecule responsible for that process remained a ghost. To identify that molecule, the scientific community had to establish 3 distinct pillars of evidence: prove its biological function, decode its chemical language, and map its physical 3D shape. This wasn't the work of a single eureka moment. It was a rigorous, collaborative effort that required researchers from different disciplines to verify each other's findings over several decades. The first breakthrough regarding function came from Oswald Avery. Through a long series of painstaking biochemical steps, Avery isolated a transforming principle from bacteria, progressively purifying the material by enzymatically destroying every other cellular component. But the results were met with intense caution. Many researchers feared that microscopic amounts of protein contamination, not the DNA itself, might be the actual source of the genetic transformation. That debate lasted until 1952, when Alfred Hershey and Martha Chase used a mechanical approach to settle what the genetic material actually was. They used bacteriophages, viruses that attack bacteria, to track exactly what enters a cell during infection. As this diagram shows, the virus acts like a syringe, attaching to the cell's surface and injecting its internal payload while leaving its outer protein shell behind. By proving that only the DNA entered the cell to direct the production of new viruses, Hershey and Chase provided definitive proof that DNA, not protein, was the functional messenger of heredity. Establishing the function was only the beginning. Scientists still had to understand how a single molecule could hold a complex blueprint. To find out, Austrian biochemist Erwin Targoff began analyzing the DNA of different species. This chart shows the composition of the 4 chemical bases in DNA: A, T, C, and G. Targoff discovered that while the amounts of these bases stayed constant within a species, they varied across different species. Crucially, the amount of A always equaled T and C equaled G. These ratios, known as Targoff's rules, demonstrated that DNA wasn't just a repetitive structural support. It was a variable system capable of carrying a sophisticated chemical code. Even with the function and the code understood, the molecule's physical architecture remained invisible. The search for its 3D shape led to the lab of Rosalind Franklin. Franklin was an expert in X-ray crystallography. In this process, X-rays are fired at a crystallized form of a molecule. The atoms deflect the rays, creating a diffraction pattern that provides the mathematical clues to the molecule's structure. The patterns Franklin captured provided the final requirement: the geometric data needed to visualize how the entire molecule was organized in space. By the early 1950s, these 3 independent lines of research, the functional proof, the chemical rules, and the structural data, were ready to be synthesized. Scientists like Watson and Crick used this accumulated data to build a physical model that satisfied every requirement, revealing the now famous double helix. This breakthrough is the direct ancestor of the modern technology we use today. Every genetic test and forensic profile relies on the architecture discovered during that era. Our modern understanding of life is the result of a rigorous scientific journey that systematically turned a historical blind spot into the most detailed blueprint in human history.